Please help me in welcoming Dr. Levings.
Thank you very much for that kind and long introduction. And thanks very much to everyone before me for setting up the stage for what I hope will be an engaging talk that convinces you that our next step is moving towards tolerance. So my lab works on, I just figure out the AV here. Oh yeah, so just I do have a disclosure that I hold some patents and license related to the CAR technology I'm going to talk about today. So my lab works on regulatory T cells, and for those of you who aren't familiar with them, they're a particular type of CD4 positive T cell, and they are essential for decreasing undesirable immune responses. If you don't have regulatory T cells, you're essentially born with autoimmunity. And over many years of basic science and translational science, we have a lot of evidence that if we could find ways to boost regulatory T cells, that would be beneficial in transplantation, graft versus host disease, autoimmunity, and other types of inflammatory diseases. So the general concept is that if you have one of these diseases or you're at risk, you probably don't have enough regulatory T cells, and so we want to shift that balance. So we have more Tregs, less of the pathogenic effector T cells that could lead to rejection. And one of the ways we can do this is by taking Tregs out of the body, expanding them in the lab, potentially introducing a genetic modification so they work even better, and then giving them back to the patient. So what I'm going to talk about today is two areas of research that my lab has been working on. In the first part, I'm going to talk about taking Tregs from the patient themselves, genetically engineering them in the dish to infuse them back as a more potent cellular product. And in the last part, I'm going to talk to some work in my lab that we're actually moving towards clinical translation, which is to use an allogeneic Treg source, so not from the patient themselves, a different source, because one of the big challenges in the cell therapy world in general is the extremely high cost and complexity of making a bespoke patient-by-patient product. All right, so what do regulatory T cells do? We like to think of them as a polypharmacy. So in comparison to the traditional immunosuppressive approach where you have one drug that does one thing everywhere in the body with no brain, it's having a systemic effect. The idea behind Tregs or any type of cellular therapy is that those cells are a biological living thing, so they can respond to the environment and react in an appropriate way. And we think that the key things that Tregs do in terms of their mechanism is that they are a T cell, so they express T cell receptors. So just like all T cells, they have to get activated by peptide MHC molecules. But then this crosstalk between the antigen-presenting cells actually results in a downregulation of antigen-presenting cell activity and physical removal of some of these co-stimulatory molecules that are important for activating other effector T cells. Then once Tregs get activated, they create a local suppressive microenvironment. They produce regulatory cytokines like IL-10 and TGF-beta. They consume IL-2, depriving other conventional T cells from that important growth factor, and they do other sorts of metabolic changes. And the overall outcome is long-lasting tolerance, where we have a combination of expansion of these regulatory T cells, as well as de novo induction of new regulatory T cells, both through TGF-beta-induced new Treg induction, as well as the development of tolerogenic antigen-presenting cells that also lead to more regulatory T cells. And in the context of cell therapy, this process of infectious tolerance is really critical because it means that ultimately it's not the cells that you infused, it's the recipient's own immune system and their new regulatory T cell pool that can maintain long-term tolerance. So that means, we think, that one injection of Tregs should be enough to induce tolerance for life. And I'm not going to show you the data today, but we have actually pretty strong evidence in mouse models that this is true. So I just wanted to highlight one clinical trial that's really been transformative for the field of Treg therapy in terms of really showing that you can use this as a way to reduce immunosuppression, because that's ultimately the overall goal here. Less of this toxic immunosuppression. And this clinical trial called the ONE study was a very ambitious multi-center, multinational trial. You can see the centers here that were actually comparing different types of regulatory cell products in living donor kidney transplantation. And so in particular, Giovanna Lombardi's group in London and Birgit Sawicki and Petra Hoffman's group in Germany were testing different types of polyclonal Tregs. And then in the US, we had Jeff Bluestone's group and Jim Markman's group testing different types of donor-specific Tregs. And the important thing is that the way the protocol was designed was that the regulatory T cells actually replaced the anti-CD25 induction therapy that these patients would normally get. So in a reference group that got the anti-CD25 induction therapy, you can see that they have quite a number of infectious events, whereas the cell therapy group, this is just all the patients together, clearly have less infection. So this is likely partially due to the fact that we have removed one of these immunosuppressive agents, but could also be due to other beneficial effects of the cell therapy. And in fact, if you look at those patients longer, you can see that in those individual studies, they virtually all allowed a tapering of the immunosuppression from triple immunotherapy to monotherapy. And so in Petra Renkre's group, basically 10 of the 11 patients achieved this after 48 weeks. And in Jim Markman's group, similarly, all the patients had been able to be tapered to TAC monotherapy. So really showing that a single injection of regulatory cells can allow you to profoundly change the immunosuppressive profile of these patients. So what my lab is working on is the next generation of those regulatory T cell therapies. And one of the things we've been focusing on for quite a while is taking an idea from cancer immunotherapy to create chimeric antigen receptor Tregs. So chimeric antigen receptors are man-made proteins where we actually take the antigen binding domain of an antibody and we fuse it to different parts of proteins that we know will stimulate T cell activation. So in this case, we add in co-stimulation from CD28 and then signaling domains from CD3 zeta. And the overall concept is that these CAR-redirected Tregs should be more potent than polyclonal cells because you've loaded them all up with a uniform specificity that should allow them to go to the site of the transplanted organ, as well as the local draining lymph nodes. And our CAR is specific for HLA-A2. And so the concept would be that you would use these cells in someone who is A2 negative receiving an A2 positive organ. So I'm just going to show you some of the highlights of the data that we've generated to support this overall concept. Just to show you how we make these cells, it's a fairly straightforward process, which would be very similar to how you make CAR T cells for cancer immunotherapy, with an important difference that Tregs are a very, very rare population of cells, and it's important to get them to be pure and homogeneous. And so that means we need to have a FACS sorting step. So what we're doing here is flow sorting, naive, CD25 positive, CD127 negative Tregs, and then we transduce them with a lentivirus so that we get nice transduced cells. The CAR is marked here by this stain, and they have high expression of their two favorite transcription factors, Helios and FOXP3. So this is in humans. We have a very similar process in mice where we can use reporter mice to sort out our Tregs, transduce them with a retrovirus, and at the end of the day, having a nice population of CAR positive, FOXP3 positive Tregs. So one of the first things we did was look at how CAR expression controls homing of the cells, because the overall concept was that the specificity should allow them to go to the local allograft. And so we addressed this by doing this experiment where we had an immunodeficient mouse that had two side-by-side skin grafts that either did not or did express the HLA-A2 mismatch. And then we loaded our Tregs with the CAR as well as a luciferase transgene, which allows them to give off light in response to the substrate, and we can actually follow that light signal through a special instrument in the animal facility. And these data really clearly showed us that the expression of the A2 CAR allows very potent and rapid trafficking of the Tregs only to the allograft. So on the top here are two mice that got the A2 CAR Tregs, and the bottom are two mice that got control Tregs, and then the bottom you can see, so the blue signal is the Tregs. It's basically petered out in the control mice. The cells don't even last long term. But in the mice that got the A2 CAR Tregs, they all go to the allograft here. They don't go at all to the adjacent non-A2 graft. they stay there, they expand and divide, and actually ultimately go out to a local draining lymph node. The other thing we did was adapt that model to ask whether they could also control rejection. So in this case, the mouse got a piece of human HLA-A2 positive skin and then got allogeneic PBMCs, which in the absence of Tregs will reject this skin. And in the presence of Tregs that have the CAR, we could see a beneficial effect on rejection. And so we measured this histologically, looking at the integrity of the little piece of skin graft. So on the top here, or on the left here, in the absence of Tregs, this skin graft is essentially destroyed. The epithelial layer in green is really not even there anymore, and we have a lot of pink Ki67 keratinocytes that are trying to heal the wound and just not able to. The control Tregs have a slight effect, which is not surprising because a certain proportion will be allo-specific, but you can see the A2 CAR Tregs are significantly better able to control rejection of that skin allograft. And this is all quantified here, both in terms of Ki67 reduction and blinded histological scoring. Now, since this is an HLA audience, I thought I'd throw in a couple of our slides on how we work to try and address the exact specificity of this CAR. So we made this CAR from a very well-known ancient antibody, BB7.2. I'm sure all the HLA people know that antibody. This antibody has been described to have some degree of cross-reactivity with other HLA molecules. Moreover, when you transition an antibody to a CAR, you can potentially change the specificity of that molecule. So it was very important for us to understand this. So BB7.2 had previously been shown to also have cross-reactivity to the highly related A69, but also at high levels, A68, A23, and A24. So in order to ask this question in the context of a CAR now, so a CAR is a protein that we're expressing on a T cell, we decided to set up this new assay that we call the flow PRT assay. Unfortunately, I'd spent enough time in transplant meetings and talking to people like Paul to come up with this idea, where we essentially repurposed the flow PRA beads, which are normally used to test serum antibodies, to test the specificity of our CAR Tregs. And so we did this by flow cytometry, where we can clearly see the beads and we can clearly see the Tregs, and then we can see the doublets here, which are the beads bound to the Treg. And because the beads have different fluorescent molecules, we can then see for which beads do we get a loss of the signal from the bead alone gate going into the doublet gate. And this is just a control here. So in white, these are non-specific Tregs. They don't express a CAR. We don't have any binding to any of these beads. But in cells expressing the specific CAR, we get a very specific loss only of the A2 peak. So we use this assay to profile our CAR, and I'm not going to go through all the data. We essentially did the whole class 1 panel and mapped the specificity of the original CAR, which is in red, as well as a number of different humanized variants that we made. And the overall conclusion is that the CAR is highly specific for HLA-A201 here. And that our humanized variants actually were even, and also for A69, which is not surprising, and that our humanized variants actually had better specificity because we had lower cross-reactivity in comparison to the original unmodified mouse CAR. And so we actually ended up moving forward with a clinical application with this particular humanized version, H1K2, which has very low cross-reactivity with any of the other HLAs that we tested. Okay. In summary of a lot of data from our lab and also a few other labs around the world, we created this HLA-A2 CAR that targets an antigen that's applicable to a relatively large patient population. I'm not going to go into the details, but we did a lot of things to minimize the risk of the genetically engineered cells. We humanized it. We precisely mapped the specificity. We showed that there's no induction of cytotoxicity. And as I mentioned, I'm not going to show the data, but we have a lot of evidence from our mouse work that they carry the properties of tolerance, important in the animal models, important for tolerance. So they have antigen-specific suppression. They localize to the graft and draining lymph nodes. And this has also been work that's been done by Elmer Yeckel and Giovanna Lombardi. And so all of this work has now led to 2 ongoing clinical trials. One is being led by a company called Sangamo, which is using our CAR in a living donor kidney transplantation context. And the outcome of this study is the tapering of immunosuppressive therapy, so very similar to what the outcome of the one study was, to try and gradually reduce those patients to a monotherapy and to reduce the amounts of tacrolimus. And the overall protocol is to, before the patient is going to have the transplant, you do a leukapheresis, manufacture the CAR Tregs, they get the transplant, and around 100 days later, they'd be infused with the Treg product and then the follow-up. The second trial is being done by a UK company, Quell Therapeutics, in liver transplantation. And in this case, they're going to be treating patients who already have a liver transplant. And these patients will take a small dose of ATG induction therapy to make space for the Tregs, and then they'll have the Tregs put in, and then they'll do an immunosuppression tapering, similar to the kidney trial. So I've heard verbal updates from both of these studies. They've both recruited around 6 patients each, and so far no big safety concerns. Some exciting histological data coming out of this study showing that they can see the cells trafficking to the graft. And so hopefully over the next couple of years, we'll get some exciting data from that. So in my next little bit, last few minutes, I just want to briefly come to the second part, which is taking this whole concept and instead using allogeneic Tregs, not from the patient. And the reason why we do want to do this is that, as I mentioned at the beginning, autologous cell therapy is extremely expensive, complex, and it would be much better if you could take one healthy source of cells and then make an off-the-shelf product that you could use to treat lots of different people. And of course, once you have a single product, it's much easier to engineer it with desired specificities, like putting in CARs, knocking out the endogenous TCR, putting in kill switches, making them less immunogenic. It just really opens up the possibilities for all the engineering you can do. So the approach that we've taken is a bit of a different, out-of-the-box approach, thanks to collaboration with Lori West, pediatric cardiologist. I'm sure most of you know her. And she had the idea that in children who are having heart surgery to correct a congenital defect, that those thymuses are being thrown in the garbage all the time. Could we use that as a source of allogeneic Tregs for therapy? And so we embarked on that project, and essentially the overall answer is yes. So here's some nice flow cytometry of thymocytes coming from a discarded thymus, lots of CD4 T cells, lots of Tregs. And some of the advantages in comparison to blood as a Treg source is it's a very, very plentiful source of Tregs. So 1 gram of thymus has 500 more times Tregs than a milliliter of blood. The cells are highly viable. They're brand new, naive, virgin cells with long telomeres. And they're actually more potent in terms of suppression of other T cells, delaying graft versus host disease, and also more stable in the presence of inflammatory cytokines. And so we've, over the last decade, worked to create a cell therapy manufacturing protocol that allows us to go all the way from a discarded thymus to a bag of frozen Tregs. And it's a quite simple picture, but I want to take you through some of the special things that we learned about going through this process. So the first thing we learned is that is the thymus, is it an organ or is it a cell? Because when it comes to Health Canada, you have to follow different regulations in terms of tissue processing, et cetera. So if you're an organ, the bar is quite low. But if you're a cell, the bar is very high. And so we actually took a hybrid approach to this manufacturing where we are following the cells, tissue, organ Health Canada guidelines for the cell isolation. So we take a thymus from the operating room at BC Children's. We work with the cell therapy lab there to dissociate those cells into a single cell suspension and then freeze them. At that point, we can do all of our sterility testing. Once that's all good, we can send the frozen product to the GMP facility in Alberta, the ACTM. And in the GMP facility, they can do the expansion. It's a 14-day process where we grow the cells in these huge bags. we remove the beads and concentrate the cells, wash them, and then freeze them. So this process is following the Health Canada guidelines for a cell therapy product and has much, much higher regulations in comparison to the first part of this process. So one of the important things we learned in this process is that it's very annoying that there's no GMP facility in Vancouver, because although Alberta is great to work with, it's still complex to transfer your product to somebody who's not close by that you can work with on a daily basis. I'll come to that at the end of my talk. So here's just a fun slide. So normally when we're working with Tregs in the lab, we're working with in sort of 50 to 100 microliter range. This is completely scaled up. So here, this is a huge cell suspension of thymocytes coming from a thymus. This is a huge magnet from Stem Cell Technologies, and you'll see the cells that are bound to the magnet going over to the side. In this case, we're depleting the cells that we don't want, and the cells that we do want are here in this clear suspension. So we'll pour them off. It's about a 200 milliliter volume instead of our 50 microliter volume. So got used to working with a completely different scale. And then one of the other big challenges was the scale of growing these cells. So instead of a few hundred microliters, we are now in 2-liter bags of cells. And in fact, the GMP facility can't handle more than 6 liters. So we actually end up throwing out cells because we just don't have a bioreactor big enough. And that's something we're working on overcoming. So what are we going to do with these cells? So our first application is actually as a trial to prevent graft versus host disease, and we were recently successful to obtain significant funding both from BioCanRx and CHR to do this study. I think I saw Depeche in the audience somewhere. Depeche is the area is at the back, is going to be the local person here in Vancouver helping us with this, so thanks so much. And this is all building upon a lot of basic science showing that allogeneic Tregs can enhance bone marrow engraftment, protect from graft versus host disease, and a lot of parallel work that's being done using other types of allo T cells in cancer and to fight infections. But there's also a fun way that you could use this as an autologous T cell product, and that's actually in a child who's having a heart surgery because they're about to get a heart transplant. This was a very difficult study to do in Canada because we don't have that many pediatric heart transplants, and so we were lucky to collaborate with Rafael Roca in Madrid, who has an ongoing study to use thymic Tregs in an autologous setting in children who are having a heart transplant. And this is just a figure from his first N of 1 patient showing that, so this little girl had a heart transplant. In parallel, Rafael made her own autologous T cells and then gave them back. And what he found is that if you tracked the number of Tregs in the blood over time, that the patient had a significantly higher Tregs in comparison to a large number of control patients who had had their thymus removed and were on a lot of immunosuppression. So it's only an N of 1, but very clear evidence that if you give these patients back some cells that you took away because you removed their thymus, you're probably giving them a more beneficial immune system. So the last 2 slides is that I mentioned it was quite challenging to transfer our technology to Alberta, to their GMP facility. So together with a large team of people at UBC for the last few years, I've been working hard to try and get funding to build a GMP facility here in Vancouver. And I just wanted to let everybody in the audience know that we have been successful in this now. And so we have funding to build a GMP facility in Vancouver that will have a multimodal capability. It's going to be able to do cell therapy, including immune cells and stem cells, make viral vectors for gene therapy, make plasmid DNA, which is the starting product for viral vectors, as well as for mRNA and lipid nanoparticles. So if there's anyone in the audience that has an idea for a therapy that they want to do, please come and talk to us, because we're in the process of buying equipment, designing the building, and really want to make sure that we have capacity and have the things that people in the environment will want to use. This will be on the UBC campus. So for those of you familiar with UBC, you may recognize this as the place where you wait for the number 99 bus in the bus loop. If you stand there now and wait for your bus, you'll see that there's a big pit of dirt here full of construction equipment. But in 4 years from now, there will be a GMP facility. It's going to be a 6-story building with the GMP facility on the top and a process development lab here, and then some other space with different functions for other purposes below. And that's just here, so you can wait for the bus and then just go upstairs and manufacture your cells. So thanks very much for your attention. It's a huge team of people who've been involved in all this work, and we actually have lots of ideas for how to work with the HLA lab, so I'm looking forward to continued collaborations.
Great progress, Megan. I'm following this with fascination over the last decade. And in the past, the problem was all the cells injected usually did die off. But with this case from Madrid, it looks like it's very sustainable, right, with the processed and refined thymus cells. There was a bit of a dip and a rebound. Is that a sign that they start reproducing after the first generation might taper off? Or how do you interpret that? Or is it too early in one patient?
I mean, I think it's too early in one patient. I guess the overall good news is that throughout the whole course, they clearly stayed, that one patient stayed higher than all the controls. So I think it's maybe not that surprising that, especially the child is taking immunosuppression, that maybe depending on slight changes in the medication or other environmental things, there might be some up and down fluctuation of the regulatory T cell pool. Yeah, but so those are autologous cells. For our product, for the thymic Tregs in graft versus host disease, it will be an allogeneic product. So we do not expect those cells to last in the long term. But because of the properties of infectious tolerance, we think that it's the host's immune system that will eventually take over.
And just one irritating thing is when you say we are making cars, me as a German, I'm thinking, that's interesting, what car? But it's the other car. No, but one lab question is really, does Health Canada in this context recognize GMP manufactured products, or do you also need to have Health Canada accreditation for your cell manufacturing when you go into clinical trials? Do you know that?
So the GMP facility will be having to follow Health Canada guidelines, and then the guidelines you have to follow depends on whether you're in the clinical trial or whether you're in a licensed product stage. So as long as it's still a clinical trial, the bar is a little bit lower, but if you want to have a fully licensed product, then it's like you're a drug manufacturer. You have to fully check all their boxes.
I think we have time for one more question.
Okay. How are they tracked in patients? So in the mouse, you can nicely let them light up and screen then. So how are they currently tracked?
Yeah, so there's two answers to that question. So if people get genetically modified Tregs, so like the A2 CAR Tregs, those can be tracked actually using HLA tetramers because the antibody binds to HLA. So we use tetramers to stain for them, and those tetramers work by flow, and they also work by histology. So it's actually a really good way to track those cells. Of course, you can also use PCR for the transgene. But for the unmodified allogeneic Tregs, I was just talking to a few people, we actually need to set up ways to track those cells, and I'm hoping the HLA lab here will work with us on that, because Health Canada, we had a pre-CTA last week, and they asked specifically for that assay, so we need to develop it.
Wonderful. Thank you so much.
Okay, thanks so much.